Alright, hello everyone. So this is going to be your final review for Biol1140. So let's see what we got here. This has been so kindly gifted to us by Nelia. This is a resource from previous years that she has gone ahead and given to us. So let's go ahead and jump right into it. So number one is asking us to label the DNA diagram. So nitrogenous base. That one's pretty easy. We have the nitrogen containing bases here in the middle. So that's these are our N bases right there. So our sugar. So the sugar is going to be a 5-carbon sugar. It's going to be this baby here. And so that's our sugar. Very good. Okay, how about a phosphate? These little circles here are our phosphates, right? Hydrogen bonds. So is this a hydrogen bond? No. Is this? No. Always with a dotted line, that's how we know something is a hydrogen bond. So what connects the N bases to each other are hydrogen bonds. Good. So the 5' end is going to be over here, and the 3' end is going to be down here. And we can go over what that means here in a second, right? So A, T, and G. So let's just fill in the blanks. So we know that A goes with T, right? So there we go. And we know that C goes with G, and again T with A, right? And then oxygen. So the oxygen is up top here on the sugar. So look good. Let's have a little bit of a closer look at as to what the nucleotide might look like, right? So this is the sugar. In this case, it's deoxyribose, right? So we have OH, and then bound to it is the N base, and over here we have the phosphate, right? So for each corner, we count 1 carbon, right? So 1 carbon, 2, 3, 4, and then we go up here. Well, that's oxygen, so that doesn't count. So we go up 5. Right, so our sugar has 5 carbons, right, and we count it going clockwise from the rightmost position. So this is our rightmost position. That's 1. So 1, 2, 3, 4, 5 carbons, right? And yeah, that's how you count it, right? So then you count the primes too, because technically the non-prime carbons are going to be here in the N base, but we don't ever care about that in this class anyway. Right, so over here we have the phosphate. Right, so the phosphate is where we attach the nucleotides. Right, so when we talk about 5 to 3, this is what we mean. Right, we have the 5' carbon and the 3' carbon. Right, so we always go down this way from 5 to 3. Right, and that goes for DNA replication, RNA synthesis, that sort of thing. Right, 5 to 3. So good. And we can see the, since DNA runs anti-parallel, we can go like that, right? So we have the 3' end over here, and the 5' end over here. Good. So what components from above make up the backbone of a DNA strand? So the backbone of anything, of an amino acid chain, of a DNA nucleotide chain, of whatever, right? The backbone is anything that's not the N bases. So anything that's excluded from the N bases, that's going to be our backbone. So that includes the sugar and the phosphate. Let me say that again. The sugar and the phosphate make up the DNA backbone, right? So we could maybe go ahead and write that in. I'm going to try to anyway, because Microsoft Word is just... it's just the worst. I'm sorry. It's just really terrible. Okay, so the sugar and the phosphate. That's what make up our DNA strand. Right, so box in 1 nucleotide on the DNA diagram. So let's go ahead and do that, right? So this is a nucleotide, right? We just drew over here. So we just have to kind of level off 1 from the rest of the group. So we could have 1 over here. That makes sense, right? Go the other way. We could also have 1 over here. Oh, not quite. 1, there we go, just like that. That's 1 nucleotide, right? For every 1 nucleotide, you have 1 phosphate, 1 sugar, 1 nitrogenous base, right? So good. So to describe the directionality of the DNA strand, well, I kind of already said it, right? Anti-parallel, meaning that, so if you take in math class, right, we know that parallel, I'm going to try and do this properly. Parallel, meaning that we have 2 lines that never intersect, but anti-parallel, meaning that they actually run the opposite direction, right? So 1 going down, and 1 going up, right? Makes sense, right? So describe how it's determined which end of the DNA strand is the 5 or the 3' end. So we kind of already described that, right? So we have 1, 2, 3, 4, 5 prime. 3' represents the bottom, and 5' represents the top, right? So we always go from 5 to 3', right? Always from 5 to 3'. And that goes for every single application, right? Except for like maybe certain minute details, no. But for DNA synthesis, 5 to 3. Got it? Good. So, moving on to cell division and cell cycle and whatnot, right? So, below is a diagram of the cell cycle. In each phase, fill in what occurs in each of those phases. Use as much detail as possible and know where DNA replication and transcription translation occur. Right. So, in G1 phase, right? In G1, that's our growth period. Our gap, G stands for gap, right? But ultimately, it does actually mean that the cell is growing and it's performing its normal metabolic activity, right? So during G1, it's just the cell is doing its normal thing. It's transcribing and it's translating. All that good stuff, right? All that stuff that we care about in the cell. So, yeah, the cell's happy during G1. It's just doing its normal, everyday thing, right? Until we get into S phase, right? So during S phase, that's when DNA replication actually does occur, right? Sorry, my cat just climbed up on my desk. I'm going to take him off. Okay, so S phase, S stands for synthesis. So what are we synthesizing? DNA, right? So good. S is synthesis. So good. So G2, right? It's kind of like G1, right? It's almost exactly like G1. But the only difference is that the DNA is now duplicated, right? In addition to this, the cell is getting ready for division, right? It's not only just performing normal metabolic activity, but it's actually getting ready for the division of the cell. Right? So that's during D2. So during G2, keep this in mind, we have scissor chromatids. The DNA is doubled, right? Yeah, that's what's happening there. So then we have the M phase, right? So during the M phase, that's our mitotic phase. So we have our 2 gap phases, our S phase and our mitotic phase. And in the mitotic phase, we have the prophase, metaphase, anaphase, and telophase. And then we have cytokinesis. And the cytokinesis is when the cell splits in half, right? And then we make 2 new cells, right? So DNA is doubled during S phase, and then during G2 phase, that's when the DNA has already been doubled, right? So notice, if you look here, we have 1 chromosome here. Oh, that's not correct. Okay, this is a little misleading. So ignore this. It should really look like this. So when we have the lines, we know that the cell has not duplicated its DNA, and we have the X's. That's when the cell has duplicated the DNA. So during G2, a little bit of more elaboration, right? That's when we have certain checkpoints, right? Certain checkpoints for the cell, saying that like, okay, you're clear for division, you're clear to do, you're clear to have as much energy as you can during mitosis. Yeah. So when these signals, when these checkpoints are inactive, when these checkpoints have been inactivated in the cell, bad things happen, right? So, a question for you who's watching the video: what happens when you have unregulation in the cell regarding division and just as much division as possible? Well, that sounds to me like cancer, right? So during G2, we have the checkpoints for the cell to go into M phase, right? We have these checkpoints that say, okay, we have this, 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 we're all good, right? If these checkpoints don't exist, then the cell's going to go into M phase anyway, and it's going to divide, and it's going to make more and more bad cells. Eventually, that leads to a carcinoma, right? Anyway, so describe the rules. Moving on, number 7. Describe the rules for synthesis of the leading strand versus synthesis of the lagging strand in DNA replication. Right? So leading strand, we go from 5' to 3', right? So that means for the lagging strand, we go 3' to 5', right? Absolutely not. No. No. What we do in the lagging strand actually is also 5 to 3, right? So let's go down here into number 8 because we already have the kind of example drawn for us, right? So we have our origin of replication, right? The origin of replication is kind of like the middle portion, right? So here's the thing, right? So we have to pay attention to where we're going. So if we're going into the replication fork, that's the leading strand. That's the leading strand. If we're going away from the replication fork, that's the lagging strand. Right? So they both are synthesized from 5 to 3, right? So we have 5' on this end, and then 3' on this end. But then, so the arrowhead describes where the 3' end is, right? So on this end we have 5' to 3', and then each arrowhead represents 3', so 5 to 3, 5 to 3, 5 to 3, right? So the lagging strand has fragments. It's fragmented. So fragments, right? And then leading strand is continuous. Continuous. So lagging strand has fragments, leading strand is continuous, right? So once we get past the origin replication, then the lagging strand actually becomes the leading strand, right? So we actually go in. Now we're going into this replication fork. By the way, this little space, as we're unzipping the DNA, this space is called the replication fork, right? Make sense? Good. All right, so we're going into the replication fork. Now this is the leading strand. And on this one, we're going away from. Right? So leading strand has fragments. Sorry, leading strand has a continuous strand, and lagging strand has fragments, right? Both of them, however, require a primer. So a primer is just a signal for DNA polymerase to go ahead and make more DNA, right? So the primer just essentially acts like a flag, right? So it's like, okay, where is it? We have a primer here, and then we just go that way, right? We have a primer here, 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 one here, 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 right? So every time we start replication, we have a primer. So therefore, for the lagging strand, we have multiple primers, or many primers. For the leading strand, we really just have one. Right? Because for the lagging strand, we have to stop and go. Stop and go. Whereas the leading strand, we could just go whoosh, right into the replication fork. Right? Good. So moving on, let's go to number 8. Right? So using the following image, complete the steps of DNA replication. Begin with labeling the origin replication of the replication fork. Then using the rule you've listed in number 6, or I assume 7, add the leading and lagging strand to the image. Next, draw how the leading and lagging strand continue to synthesize. Lastly, draw what the final products look like. Okay, cool. Right, so these sort of green squares is going to be our origin replication, right? And like I said, as the DNA is being unzipped, we have our replication forks, right? And we have our lagging and leading strands, right? So once we pass the origin replication, the roles reverse, and the lagging becomes the leading, and vice versa, right? So once we're done, we have a DNA strand that kind of looks like this, right? So we have two identical, and keep that in mind, two identical strands of DNA. And remember, they have to run anti-parallel to each other, right? Just like that. Simple enough? Good. So, here's the thing. So this is happening during S phase, right? This is happening during S phase, right? One more thing. We are not, and pay attention, we are not duplicating the chromosome number. We are not duplicating the chromosome number. What we are duplicating is the DNA. But we are not duplicating the chromosome number because, let's see, if we're in G1, we have like two chromosomes that look like this, right? And then we go through S phase. Now after S phase, we're in G2, right? And now our chromosomes are going to look like this, right? So this is a pair of chromosomes. We can say these are homologs of each other, right? Meaning that they are similar length, similar content, different overall. They're not identical, right? So we can say this is a pair of homologs, right? Homo meaning same, right? Log meaning logic. So these are homologs of each other, right? They're not the same, but they're similar. This is one chromosome, and this is one chromosome, right? Sorry, one sec. So once we go past S phase, now we've duplicated. Now this is one chromosome, and this is one chromosome, right? So we have not duplicated the chromosome count. We've only duplicated the DNA. To duplicate the chromosome count, we have to make unique DNA. And in this process, we are not making unique DNA, right? So we have one chromosome and another chromosome. This is still a homologous pair, just like this is a homologous pair, but they've just been duplicated. So we have this sort of X conformation, right? So we have one sister chromatid and another sister chromatid to make one chromosome. And in a pair, we have two homologous chromosomes, right? So diploid cells have homologous chromosomes, right? Meaning they have two sets of genes. When we say something has two sets of genes, it just means that they have homologous chromosomes. Like I'm sure you've heard that you get one set of genes from dad, one set of genes from mom, right? This is the reason why. So, good. To reiterate, during S phase, we are not duplicating the chromosome count, but we are duplicating the DNA. All right, moving on. So, number 9. Use the following images to describe the steps of transcription. So first, label the promoter DNA. Next, and the terminal DNA, and the RNA polymerase. And then beginning working through each step, adding complementary nucleotides, and blah, 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 blah. Yeah. So, we got that, right?
So, here's the thing. This is our RNA polymerase, right? This sort of yellow circle, RNA polymerase. And just from this image, green tells us go, red tells us stop, just like, your typical traffic light, right? So the promoter will go ahead and be this green portion, right? So this is our promoter DNA. Right, so promoter, like literally, what does a promoter do? Like, I imagine, like, I live right next to this coffee shop that has like a guy standing outside sometimes saying, like, waving a sign like, hey, come drink our coffee or whatever. That's what this means, right? A promoter is just a region that tells the RNA polymerase where to go, right? Just like how that guy with the sign is telling me where to go drink coffee, the the promoter DNA is telling where the RNA polymerase should go, right? So that's initiation, right? So we have the RNA polymerase that attaches at the promoter DNA and then attaches to the DNA. So now it's in it, it's unzipping the DNA, so it's unzipping like the portion that it needs to access, right? And as it's doing this, it just kind of unzips, and then it gets zipped back up, right? Kind of how you see here, right? So that's initiation, just where the RNA polymerase attaches, right? So now elongation. So to elongate something means to make something longer, right? So as we're going along transcription, right? So transcription, if you're taking notes, which I hope you are, you don't have to, but I hope you are. If you're taking notes, you are transcribing something, right? To transcribe means to literally, it literally means across writing, right? So transcribing something means that you're taking a source and you're writing directly from that source, right? So that's what we're doing here. So the RNA polymerase is kind of, as we're unzipping, we have our base pairs, right? And and the RNA kind of just leaks out like that, right? It's going to be our 5' end, it's going to be our 3' end. You don't need to know that detail, it's just how it is, right? So as we're going along, as we're elongating, as we're matching base pairs, we are making this mRNA longer, right? So once we've made our mRNA long enough, we hit the terminator region. So in the terminator region, like in our analogy of stoplights, it just tells us to stop, right? It literally just tells it to release from the DNA, and now they're separated. So now we have an mRNA, it's a 3' and a 5' end. We have our DNA, which is completely unphased, just normal, ordinary DNA, ready for another RNA to be made from it. And then you have our RNA polymerase, which is released, right? Really simple, right? So initiation, elongation, termination, right? That's all that there is to it. Initiation at the promoter, right? RNA polymerase attaches. Elongation throughout the DNA. We read the DNA. We're making RNA, and then termination, right? We have termination where we reach the terminator DNA sequence, and then we just stop, right? Simple as. So the template strand of DNA reads AGT, CT, AGA. So what would the complementary RNA strand read? Well, let's find out. You don't need to know this detail, but this DNA would read would read from 3 to 5. Right? So A, G, G, C, T, A, G, A. Right? And then 5'. So our RNA is going to read the opposite, right? So for DNA, A pairs with T, and then C pairs with G, right? That's for DNA, for base pairing DNA. For RNA, A pairs to U and C to G. So, with that in mind, A to U, G to C, C, G, A U C U. Right, so U C C G A U C. Right, and maybe I can yeah. Just like that. That's how it should read, right? So, yeah, that's really all there is to it, right? So A to G, A to T in DNA, A to U in RNA. That's the only difference, really. Good. So number 11, during translation, the blank on the tRNA molecule. So now we're in translation, right? The blank on the tRNA molecule binds with the blank along the mRNA strand, right? So here's the thing. For this, we have to know the anatomy of a tRNA. But for now, let's just answer the question, right? So the anticodon, I know for a fact that Word is going to give me a little error on this spelling. Or not. Maybe I don't know for a fact. Okay, so the anticodon on the tRNA molecule binds with the codon along the mRNA strand, right? So for this, we have to know a couple of different words, right? So tRNA, I'm just going to draw it down here, right? So tRNA quite literally looks like a T, right? So T stands for transfer or translating. Kind of looks something like this. And I did not want that to happen. Wow, incredible. Okay, kind of looks something like this, right? So this is just one long RNA strand, and then we have a sort of sequence down here at the bottom, right? So this sequence down here at the bottom is going to be our anticodon, and this little bead on top is going to be the amino acid, right? So what the anticodon is it binds to a codon, right? So we have the most popular example of a codon, AUG. That's our start codon, right? So AUG exists on the mRNA, right? So 5' to 3', right? AUG exists on the mRNA. So what's going to happen is that the anticodon is going to read UAC, right? So the anticodon just essentially base pairs with the codon. The codon being a 3-letter sequence or a 3-base sequence that helps the tRNA recognize and bind to, right? So this is how we elongate our amino acid chain. We bind, and then the amino acids kind of do a little sort of transfer, and then they make the chain longer and longer and longer and longer, right? So that's our anticodon, a UAC, and our codon is AUG, right? Codon being on the mRNA, and the anticodon being on the tRNA. Good. So, use the following images to describe the steps of translation. Okay, so it's asking us to name, to add the ribosomal sites, to add a brief description of them, happens at what each step, and what each component is, right? So this is our tRNA, right? So the tRNA has a little bead, and it has the anticodon, the three-letter anti-sequence to the codon. We have the mRNA, and we have the ribosome. So now the ribosome has 3 sites to it: the A site, the P site, and the E site. So the A site, so here's the thing. I kind of came up with my own definition. You could use the definition from your notes. Totally fine. They kind of mean the same thing, right? A site is acceptor site, right? So we are accepting the tRNA, right? So the tRNA is being accepted here, and the P site, I like to say, it stands for processing. For me, anyway. Other people might say it stands for protein or peptidyl or peptide, what have you, right? So A to P, and then P to E. E stands for ejection or exit, right? So that's where the tRNA is ejected from the ribosome, right? So initiation, right? So to initiate translation, we need the start codon, right? So we need the start codon to initiate translation. Of course, the start codon is AUG, which codes for methionine, and we need the ribosome. So what happens here is when we have AUG, so right there on the mRNA, right at the beginning, or, well, really, it would be right here. Doesn't matter. That's too much detail for this class. So right at the beginning, right at the start, we have AUG. So AUG, of course, is a codon, right? So for our first codon, we actually bind here at the P site, right? We actually bind at the P site. It's an important detail, right? Binding at the P site. For initiation, anyway. For our start codon, that's where we bind. So what's not drawn here is that the ribosome comes in two parts, and the two parts come together during initiation. We don't need that level of detail, I don't think. But we do need the start codon, and we need the ribosome, so the tRNA goes ahead and binds at the P site. And then we go to elongation, right? So then during elongation, we have another tRNA that comes in and binds at the A site, the acceptor site, right? So what happens next, right? What happens next is we have this amino acid making a chain with the amino acid in the P site, right? And then everything kind of shifts to the right. Sorry, the ribosome shifts to the right. Technically, everything shifts to the left, right? So we're shifting over. So now what's in the P site moves to the E site, and what's in the A site moves to the P site. And then we have sort of this, right? So we have our first amino acid is down here, right? And our next one is up here, right? So during elongation, we have a tRNA at the A site, and the amino acid attached to the tRNA gets attached to the amino acid at the P site, right? So we're attaching here, and now this tRNA is at the E site, which means it's going to eject. It's going to leave. It's going to exit, right? And then we just continue this process, right? And then another tRNA gets added to the ribosome, right? We have another tRNA. It's at the A site, and it's the same process. This amino acid goes ahead and attaches. This amino acid attaches to the other amino acid, and then the process continues and goes on and goes on, right? So that's how we make an amino acid chain, right? So the pentagon always kind of starts here because that's where we started first, right? And then this entire chain is going to kind of move, right? So we have this one here at the A site, which is going to attach at the very top, right? And yeah, it's just details at that point, right? How it works. Anyway, moving on. So termination, right? So we have termination. So the thing about termination is exactly that. We're terminating the sequence, right? So we have this new special type of tRNA that codes for a release factor, right? So a release factor essentially just tells the ribosome to release the amino acid chain to release the mRNA and to split apart the ribosome, leading to something that kind of looks like this, right? So here's the thing: the stop. So what codes? So this right here is technically actually a stop codon, right? So that's a stop codon, but here's the other thing, right? Stop codon does not code for an amino acid. Does not code for an amino acid. Does not code for an amino acid. It codes for the release factor, which tells everything to come apart and release, right? So what kind of reaction does it cause? I believe it's a hydrolysis. I could be wrong in that, but I believe it's a hydrolysis. I would advise looking that up if you're so inclined to. So, that's termination, right? We start initiation, right? The tRNA attaches at the mRNA attaches at the ribosome, and then the tRNA attaches at the mRNA, and then the other ribosomal subunit attaches, and then we're at the P site, right? So we start the P site. We don't start at the A site, we start at the P site, right? And then elongation, we have another tRNA that comes in, attaches its amino acid at the A site, and then everything shifts to the left as the ribosome is reading to the right. Everything is shifted, so now what was in the P site gets in the E site, and what's in the A site gets to the P site. So whatever's at the E site is then ejected, and then the cycle continues until we hit termination, where we have a stop codon that will attach at the A site, which codes for a release factor, which tells the ribosome to let go of everything to perform a hydrolysis with the amino acid chain, where it will then be processed. I would ask any questions, but of course I can't ask that through the screen, right? So that's translation and transcription, right? One thing you'll notice is that translation and transcription actually have the same 3 steps. Now they differ, sure. They differ in the actual details of what happens during each step. However, transcription has initiation, elongation, termination. Translation has the same 3 steps. So something to keep note of. Moving on. So, number 13. Describe how people can remain lactose persistent into adulthood. Right? And so lactose persistent, right? So we have to learn what that means, right, first. So lactose persistence is the same as lactose tolerance, right? So how do adults remain lactose tolerant throughout adulthood, right? So here's the thing. I was gonna okay. Anyway. So to all of you who can drink milk, you are technically the result of a mutation, right? Myself included. Normally, adults cannot drink lactose, right? It's weird. It's really kind of strange that humans drink cow's milk because we're drinking the milk of another animal. That's very unusual in the animal kingdom, right? So, so here's the thing, right? Lactose persistence into adulthood is the result of mutation, right? So your parents have been mutated at some point because their parents are likely also lactose tolerant. So yeah. So here's the thing. So we have a mutation in the parent, right? So that's when. So this mutation codes for lactose, the lactase gene. Right, so lactase is the enzyme that breaks down lactose, right? So we have a mutation in the parent, which then has to result with a child with a mutation. Right? So the mutation is could be wrong about this, honestly. I'm just kind of, for this question in particular, I'm just kind of winging it as we go along. so, so, so yeah, you will probably have to fact check me on this one. I'm just trying to like work through it logically. So the mutation is from recessive to dominant, right? So we have a mutation in one gene, right? From recessive to dominant, which means that the mutation is much more likely to appear in someone, right? Right. So okay. So here's the thing. So yeah, the mutation is from recessive to dominant on chromosome 2. So you have 23 pairs of chromosomes. Humans do, so with a total of 46. So on chromosome 2, you have this mutation from recessive to dominant for lactose persistence, right? So therefore, a child with a mutation will transcribe into adulthood. It's transcribe an mRNA, which will then be translated into lactase. Right? So a mutation, here's the thing about mutations. Mutations have to, I mean they have to occur in the DNA. Right? That's the thing about mutations, right? So mRNAs can change, proteins can change, they could be altered, but that's just one mRNA, one protein, right? If you change the DNA, if you change the fundamental instructions, that's when stuff gets bad, right? That's every single mRNA and therefore every single protein after will be altered, right? So a mutation from recessive to dominant on chromosome 2 for lactose persistence had to originate from a mutation in the parent, which then gives the child with a mutation, which gives the child the ability to transcribe mRNA, which gives the child the ability to translate that mRNA into lactase, which gives the child the ability to break down lactose. Good.
Hi, Ethan from the future here. I'm here to properly explain numbers 15 and 14, right? So these are both two graph questions. So for number 14, we have a graph that reads blood glucose levels on the y-axis and time in minutes on the x-axis, right? So we have two lines, line A and line B, right? So we're trying to determine which one of these is lactose tolerant versus which one isn't, right? So line A represents someone who is lactose tolerant because lactose has been digested, which means that lactose has been broken down into its monosaccharides, glucose and galactose, which therefore leads to a glucose increase in the blood. So that means glucose content, blood glucose levels have been increased as a result of this. Right, so line A, we see a steady incline of glucose, and then a slow and steady decline towards the end. Right, so line B tells us that, well, the blood glucose levels remain constant, right? So there was no lactose that was digested, right? So we just have the lactose. It's not being broken down or anything, so there's no more glucose that's being formed, or no more glucose that's present as a result in the blood, right? So moving on, number 15, we have another graph. This time it's a hydrogen breath test. So on the y-axis we have hydrogen breath levels. On the x-axis we have time in minutes. And we have the two lines again, right? Line A and line B. Line A, this time, is going to tell us, once again, lactose tolerant, because as a result of lactose being broken down, hydrogen is formed, right? So lactose being broken down, hydrogen is formed as a result, right? Obviously, line A, because we see the hydrogen levels increasing and then steadily decreasing later on, versus line B, which is a constant amount of hydrogen, which tells us that no lactose is being broken down.
So, yeah, moving on to number 16. Okay, so number 16, right? So, kind of ignore what I did here at 14, 15. This is a mistake, and I have to go ahead and I have to go ahead and edit it out and include the correct answer, right? So, number 16 is asking us for the difference between mitosis and meiosis, right? So, the function of mitosis, right? So, you're an adult person, assuming you're watching this and you're in this class, right? And if you're not, um, I'm I'm surprised. But anyway, right? So you originated as one cell, is what I'm trying to get at. You originated as one cell. So that cell had to grow and grow and grow into millions and possibly billions of more cells, right? So, how did it do that? Through mitosis. So, growth, organism growth, and to make more of itself, or to make more cells, right? So for growth, to make more cells. That's the point of mitosis. It's really simple, right? Just to grow as much as you can through cell division, right? So meiosis is solely for the purpose of sex cell production. That's it. Solely for sex cell production, which means sexual reproduction. Right, so mitosis produces every single type of cell in your body, right? So you have the rapidly dividing ones like skin cells or stomach cells, and you have the ones that don't divide at all, like skeletal muscle cells or your brain cells, or nerve cells rather. Versus meiosis, which is solely for sex cell production, which is therefore for sexual reproduction. So a cell type made for mitosis is going to be a diploid cell, diploid somatic cell, and the cell type for meiosis is going to be haploid gamete cell. The chromosome number is 2n equals, let's give the example for humans, 2n equals 46. And the chromosome number for meiosis is going to be n equals 23. Right? Good. So we can go over what that means. Right? So remember what I said, right? N, so a diploid contains two sets of chromosomes. That's what the 2n stands for, right? Di means two, so two sets of chromosomes. 2n equals 46, which means we have two sets of 23 chromosomes. One set from dad, one set from mom, right? 2n equals 46, meaning that we have 23 chromosomes times 2, 46 chromosomes, which means that we have homologous pairs, right? So chromosome 1, for example, so if we have chromosome 1 and chromosome 2, we have this pair, and then we have this pair, right? So notice how so these ones are similar, and these ones are similar. So these ones are homologs, therefore, and these ones are also homologs, right? So, yeah, that's pretty much it. So for meiosis, n equals 23 means that we only have 23 chromosomes. So you don't have one from dad, one from mom. No, we only have 23, right? Meaning that we don't have the homologous chromosomes. So it looks something like that, right? So we have one big, one small. Not too big, too small, right? No, we don't have the homologous pairs, right? That's the only difference. Gametes have half the amount of chromosomes that normal cells do, right? So the location in the body, mitosis happens everywhere. meiosis happens solely uh, in men it would be the testes. In women it would be the ovaries. Right, that's where your sex cells are produced. Okay, good. So moving on, using the following key, list both the phenotypes and possible genotypes for each of the red blood cells. Okay, so we have the green and we have the blue. Blue is type A blood, green is type B blood, right? So if we have the presence of blue antigens, that tells us that it's type A. The presence of B antigens, that's the presence of, type B blood. If we have none, then that tells us we have type O, right? And it also, what that tells us about the genotype is that we have I, nothing else. That's all that could happen, right? So the thing about blood type is that we have the gene that carries the code for blood type, right? And we have this gene, I capital A, and this gene, I capital B. Right? And we have a secret third one where it's recessive, which is lowercase I, right? So these are going to be co-dominant to each other, and this one's going to be recessive, right? So that's why you see AA over here and then AI, right? So AI tells us that we have a dominant gene for type A blood, and we have a recessive gene that's not represented, right? So for type O, we have this. Pretty simple. So here we have just the type B antigens. So of course it's pretty obvious, type B blood, which tells us that we have either two types of genotypes, right? Looks like that, right? IB, or IBI, right? Because once again, the lowercase I is not represented because the antigen is not. There is no antigen for type I blood. Therefore, it's recessive. Right. So for this one, right, we have both A and B. So we have type AB. Right. Pretty straightforward. And we can only have one possible genotype for this as well. Right. IA. IB. So both of these are going to be expressed. Both antigens are going to be expressed, right? So, yeah. Down here we have the genotype. Up here we have the phenotype. The phenotype is the actual physical trait, the physical manifestation of the gene, and the genotype is what reads in the genes, right? So the two letters or the two sets of genes represents your chromosomes, right? So on one chromosome you might have a dominant allele. On the other chromosome you might have a recessive allele. Whichever one's expressed determines dominance or recessiveness or determines the phenotype, right? Forget what I just said. What's shown in the genotype determines the phenotype, right? So down here, for type A blood, we have AA or AI. Over here for type B, we have BB or BI. For type O, we just have II, right? Because II is going to be recessive versus dominant. Dominant is always going to be expressed more often than recessive is, unless it's incomplete dominance, which is a topic for another day, right? So fill out the rest of the Punnett square regarding tortoiseshell and calico cats, right? So we have, over here we have XO. So this is telling us this is an orange male cat. Right here we have XBXB, which tells us this is a black female cat. Right, so we fill out the rest of the Punnett square, right? So we have a chance for 2 black male cats, or we have a chance for two calico female cats, right? So 50% of the offspring will be, so 50% will therefore be calico, and the other 50% will be black male cats. Calico female. Right, so 50-50, right? This is a Punnett square. So Punnett square is pretty simple, right? It kind of just works like these division squares that you might have done as in elementary school. So let's say we want to multiply 12 by 12, right? So you just take the constituents, so 10 and 2, so you multiply 10 by 10, so that's 100. Multiply 10 by 2, that's 20, right? Multiply 10 by 2 again, that's 20. And multiply 2 by 2, which is, whoops, which is 4, right? So we get 144, bring out your calculators, or if you just remember your times tables, that's going to be the correct answer, right? So same concept, right? Let's say we're trying to cross, whoops. So we're trying to cross like capital A, lowercase a, and then capital A, lowercase a, right? So we just take the constituents, right? So this is one parent, this is the other parent. You just take the constituents, and you just multiply across, right? So capital A, capital A, lowercase, capital A, lowercase, lowercase, lowercase. So we have 75% that will be a dominant phenotype, and 25% that will be a recessive phenotype, right? That's pretty simple. So there was your final exam review. This is just from the some of the resources that Nalia has provided for you. So yeah, if you are watching to the end of this video, I hope you have a good night or a good day or good whenever you're watching this, and I hope you do well in your exam. So take care, and I hope you do well. Bye.